Hello, my name is Tatiana Kuzmenko. I am one of the instructors who teaches General Biology Laboratory at Loyola Marymount University. I'd like to present here at-home laboratory activity in which students culture yogurt to explore microbiology concepts that we developed during the remote instructions in 2019. We selected milk fermentation for our remote lab for two main reasons. Firstly, by studying microbiology from the food fermentation standpoint, students can develop deeper appreciation of the role of microbes in our everyday life, so they are more interested in the subject. Secondly, milk fermentation allowed us to explore microbiology without special lab equipment. Lactobacteria breaks down sugars in milk into lactic and acetic acids, causing lowering of the pH that triggers milk protein to aggregate, forming yogurt gel-like structure. Other factors affect yogurt physical qualities as well. All this allows students to evaluate microbial activity by measuring physical properties of yogurt, such as texture, viscosity, and smell. Notice that students were instructed not to taste the yogurt they made to avoid any health risks. Students worked in groups 3 to 4 over Zoom. After learning how to make yogurt at home and evaluation of the preliminary data, a group would formulate a testable hypothesis about the conditions necessary for yogurt fermentation process, such as temperature of fermentation, time, type of milk used, or choice of yogurt starter, and design a unique experiment to test it. Many groups design their own methodology, an example of which will be highlighted in this poster. You can find the details of those assays in the paper attached below. Let me introduce our student presenter, Jackie Raetz-Vigon, with her project titled Increased Viscosity of Greek Yogurt Inoculate among yogurts containing Streptococcus thermophilus and Lactobacillus bulgaricus.
Hello, my name is Jacqueline Raetz-Vigon, and I investigated microbiology concepts using an at-home yogurt making experiment under Professor Kuzmenko. In our experiment, my group asked whether different yogurt starter types, all containing the bacteria Streptococcus thermophilus and Lactobacillus bulgaricus, would create different inoculate viscosities. First, let's define some important terms and concepts. Yogurt is generally used to mean a dairy product that is fermented by S. thermophilus and L. bulgaricus. S. thermophilus is one of the most commonly used bacteria in yogurt. Interestingly, it can produce energy regardless of oxygen presence. As well, it normally has no negative effects on its host. However, if the host's immune system is extremely compromised, it could harm the host. L. bulgaricus is also very common in yogurt, but despite its evolutionary progress, it is still not very efficient in the milk environment because it cannot break down lactose on its own. All of the yogurt starters that we tested contained these two bacteria, but they were present in different ratios and with different additional bacteria. We hypothesized that these differences, as well as the differences in production between different types of yogurt, could cause differences in our homemade yogurt viscosity. Our prediction was that yogurt made from Greek starter would be the thickest. In order to test this, we made yogurt by placing 2 tablespoons of the yogurt starter into a container of milk that had simmered on the stove for 2 minutes. This was repeated for each of the 4 yogurt starters: plain, vanilla, French, and Greek. The inoculate was then incubated in the oven with the light on for 8 hours and subsequently refrigerated for 24 hours. Each group member did this, providing 3 biological replicates per starter type. To test viscosity, we conducted the splat test. We held a level spoonful of yogurt at a height of 2 feet and 5 feet and allowed the yogurt to drop. We measured the diameter of the splat at the widest point. The wider the splat, the less viscous the yogurt. We found that the 2-foot splat test was more reliable than the 5-foot splat test. From 2 feet, it was easier to prevent human error, while at 5 feet, we had more difficult time being precise with our drops. Quantitatively, you can see that the standard deviation of the 5-foot splat test is larger than that of the 2-foot splat test, demonstrating the 2-foot splat test more reliable precision. Because of this, we analyzed our 2-foot splat test data. Using an analysis of variance test, we found that all yogurts were significantly different with the yogurt made from Greek starter being the most viscous, followed by that of the vanilla starter, French starter, and plain starter. We wanted to understand why these different yogurt starters produce different inoculate viscosities. So we first looked into bacterial differences. In the scientific literature, we found that different types of bacteria can in fact change the thickness of yogurt. According to one study, L. acidophilus increased viscosity the most, but combinations including L. bulgaricus actually tended to decrease the viscosity of the yogurt. In another study, the yogurt that fermented the fastest was made up of S. thermophilus, L. bulgaricus, and L. acidophilus, with the yogurt that was made of S. thermophilus and L. bulgaricus not far behind. So we tried to see if these applied to our experiment. We analyzed the types of bacteria in each type of yogurt and found trends inconsistent with what we read in the studies. Firstly, all of our treatments contained S. thermophilus and L. bulgaricus. So the fact that all the consistencies were significantly different would not make sense if the bacteria were the main cause of the difference. As well, L. bulgaricus was the primary bacteria in our second most viscous yogurt and the secondary bacteria in our most viscous yogurt. So L. bulgaricus could not possibly be the culprit causing the lower viscosity in our yogurts as the studies would suggest. As well, the yogurt that contained S. thermophilus, L. bulgaricus, and L. acidophilus, plain, was not the most viscous as the literature implied, but was in fact the least viscous. For all these reasons, we do not believe the bacteria themselves are the main cause of the difference in viscosity. We then considered production differences. While this could account for the differences between Greek and the other yogurt types and French and the other yogurt types, it cannot account for the difference between plain and vanilla yogurt, which go through the same production processes. Fermentation could have factored into the difference in yogurt viscosity, but it was not the only factor. One study showed that higher sugar concentration was correlated with a higher viscosity. This seems very valid since the bacteria break down sugar as a source of energy. As implied by the Dahlin study, this breakdown creates lactic acid, which in turn thickens the yogurt, among other things. We analyzed our data, finding the grams of sugar per ounce of yogurt. This accounts for the relatively low viscosity of plain compared to the other yogurts, specifically vanilla. Production differences could have accounted for all but the difference between plain and vanilla, and the amount of sugar fills in that gap since there's almost 2 times the amount of sugar in vanilla yogurt as in plain. In conclusion, we thought that a combination of production, sugar content, and bacterial differences caused the variation in viscosity in our experiment. Thank you.